Okay, I think we are 3 minutes into the hour, so I think we'll start. Please allow me to welcome our speaker for today, Professor Mark Cooper. This is not Mark's first time talking to us during this COP, but I would like to introduce him once more for the people who are joining us for the first time. Professor Mark Cooper is from the University of Queensland. He is the Deputy Director for the Center of Excellence for Plant Success in Nature and Agriculture. His main research interests combine theoretical simulation and experimental investigations to formalize the use of prior knowledge of trait genome to phenome architecture to design breeding strategies that can deliver accelerated crop genetic improvement for quantitative traits. He has breeding experience in both academic and private sector. And Mark also is a big partner with the CGIAR as part of the breeding program assessment and works a lot with our program. So Mark, you're very welcome, and I'll hand over to you at this point.
Thanks very much for the very generous introduction, Dorcas. I am very happy to be invited back. I guess that means that I didn't do too badly the first time. Dorcas asked me to talk a bit about managed stress trials in breeding, and so I was happy to do that. It sort of allowed me to sort of reflect on a bit of the history of work that I've been involved in over the years. So what I'm going to do is not sort of get into a lot of specifics about each program, but I'm going to cover some background from my wheat breeding experience. And this was collaborative work that actually started as my honors project when I was an undergraduate student at the University of Queensland, connecting and working with CIMMYT, and then ultimately becoming a wheat breeder in Australia over this period, 1985 to 2000. And then I'll also talk a bit about my experiences on trying to develop and apply managed environment capabilities to complement on-farm testing. While I was a maize breeder with Pioneer Hybrid based in the US. And I started collaborating while I was still at the University of Queensland on some of these technologies starting in 1996 and joined there full time until 2017. Pioneer is now part of Corteva, and I continue my interaction with Corteva, and Corteva are one of the key partners of our Center of Excellence that Dorcas mentioned. So what I'm going to do is I'm really just going to focus on one question that I think is maybe of interest and probably the basis behind why Dorcas asked me to talk a bit about my experience with managed environments, and I mean, try and give you some information from my own experience on how to and answer the question, can we actually use managed environments to improve the alignment of our testing system with our target population of environments? And in the previous invite to speak to this group, I talked a bit about work that myself and a lot of others have been doing over the years on trying to help people to understand how you can extend the breeder's equation that people often refer to not only think about predictive accuracy in the training data sets that we actually develop from breeding programs or designed experiments, but also to think about how to align those with your target population of environments to ensure that the genetic gain that you're realizing in the on-farm target population environments is utilizing trait variation that's relevant for performance in those target population environments. So I'm sort of going to use examples where managed environments is a technology that can be evaluated within this framework. And then around this, to take advantage of improving our understanding of the genetics of traits and how to phenotype, you can actually think about within this framework, if you design it adequately, it gives you a really good resource, data resource, and experimental resource for applied phenomic technologies using your genomics with the phenomics for mapping and building training data sets for genomic prediction and understanding environments better to try and tackle some of the questions that a lot of people are working on in dealing with genotype by environment interaction and prediction. So one of just the historical context that might be interesting for people to appreciate. For those of you that are familiar with the history of managed environment testing at CIMMYT, so a number of you might be quite familiar with that. One of the early proponents of this was Ken Fisher, who was a postdoc at CIMMYT working in the Maize program. And he actually started with other colleagues there back in the late 1970s, working on developing the managed environments that are actually have been used for Maize at CIMMYT. And Ken Fisher actually employed Greg Edmeades. And some of you may be familiar with Greg Edmeades, and just to sort of build out this pedigree a little bit, Greg Edmeades employed as postdocs Renee Lafitte and Marianne Banziger. So a number of you may be familiar with both of those individuals. So Ken Fisher, who, as I mentioned, employed Greg Edmeades, actually became the crop physiology professor at the University of Queensland when I was an undergraduate student. So he actually was part of the supervisory team of my PhD. So I was sort of introduced to this whole idea as an undergraduate student studying plant breeding at the University of Queensland, and it also featured quite a bit in the work that I was doing with my PhD. One of the other interesting connections that I may forget to mention, but I'll mention it now, is that when I joined Pioneer full-time in 2000, Greg Edmeades had already joined Pioneer, and so I was fortunate that I could work directly with Greg Edmeades on building the managed environment capabilities that we actually used for the maize breeding program I'll talk about. So it's a little bit of context in this, that like a lot of things in science and research and plant breeding, there's a large network of collaborators that you interact with and rely on and continue to collaborate with. And that sort of just gives you a little bit of an intro to the history of how I was introduced to these ideas and how they then started to feature in some of the research I was involved in and applications of these technologies in breeding. So moving on, I'll start off with a little bit about some of the work that we did in Australia to support the wheat breeding work that I was actually doing. And what I'm going to do is just talk about, based on a long series of experiments we were running where we were trying to understand our target population environments whenever we were running the breeding experiments that I was responsible for. We spent quite a bit of time trying to characterize these environments. And one of the strategies that we worked on, as well as collecting a whole lot of environmental variables for describing our environments, was using those environmental variables in combination with crop growth models to integrate those variables over a time series of crop development to look at some of the integrated variables, such as the demand that a developing crop canopy put on water resources. And so this would be the water demand for a canopy to meet the transpiration demands of the environment. and to relate that to the water availability. So we could actually look at the water supply, availability of water, whether that was through rainfall or rainfall and irrigation relative to the crop demand. If the water supply was adequate to meet the demand of the crop canopy, then that ratio would be 1. As water availability diminished, then water supply would start to fall relative to the demand. And you could think of this as a stress index. And if you went down to 0, it would just indicate to you that there was no water left to meet any of the transpiration demands of the crop at any point during crop development over the cycle. So in this example here, we integrated a lot of these environmental variables with a lot of soil characterization to look at the availability of water relative to flowering time. So this 0 point here, just for a reference genotype or a maturity group, you could think of this as, would represent flowering time. And then you could look at the water supply demand ratio before, around flowering, and after flowering. So you could start to characterize your environments in terms of whether they were water sufficient, so there was enough water available through flowering time and into grain filling, or whether they were water deficient and we had a pre-flowering to flowering stress, or a flowering stress that continued into grain filling. And in some cases, if we had a rainfall event, that would be relieved during grain filling, so you'd have mostly a flowering stress, but not a grain filling stress in this case. So this would describe these environments. So these were things that we would talk about as environment types, and they didn't necessarily represent every single situation that you might encounter, but they gave you a broad characterization of types of environments that turned up with a reasonable frequency over the years and locations in our target population of environments. Then within those experiments, what I was interested in was if these different environments were sampled in my multi-environment trial, did they give rise to genotype by environment interaction? And was that genotype by environment interaction strong enough that it could actually change the ranking of genotypes between environments and diminish the genetic correlation between perhaps a water sufficient or low stress environment and a water deficient stress environment? So then we started to study comparing a lot of these environments, what happened to the genetic correlation in the different combinations of managed environments that we were setting up. So on the right-hand side here, what I've just created is a response surface that's empirically generated over about a decade worth of wheat experiments, where we were looking at these different environment types. So this environment type 1 here would be the water sufficient environment type. And environment types 2 and 3 would actually be the water stress environment types. So what we're following on here is the frequency of environment type 1. And so as environment type 1's frequency goes up, then the frequency of the environment type 2 and 3 would actually go down. So we're just following this frequency with which we'd sampled environment type 1. And you can do this in the on-farm target environments where we were doing this characterization. And then we could also do this in our multi-environment trials. So on both of these axes, you can think of them as continuums of environment types 1 and also environments type 2 and 3 together. And so if you had an example where in our multi-environment trial, we only had environment type 1, you'd be sampling environment type 1 at this very high frequency up here. Someone's drawing on my screen, so I'll probably see if I can get rid of that, but I can't. And then what you'd be interested in is comparing the the multi-environment trial to the target population environments and asking the question, is there good alignment of the frequencies of occurrence of the environment types in your multi-environment trial and your target population environments? So in this case, there would be a very good match if the frequency of environment type 1 was 1 in the multi-environment trials, and also if it was 1 in the environment, in the target population environments on farm. And in that situation, we would end up in this back corner here where we would have a high genetic correlation of this coefficient here would be approaching 1. In this case, it was around about 0.9 between our multi-environment trial and our target population environments. But what you can also see is that if you're in the situation where you're undersampling environment type 1 in your multi-environment trial, you can start to move away from this back corner and start to head towards this situation down here where you're actually undersampling environment type 1 in both the multi-environment trial, and it doesn't occur with a high frequency in the target population environments. So what I actually found when I started evaluating a lot of the testing system that we were doing for wheat in the region I was working on in Australia, that we were often undersampling this important environment type. So a lot of the time we were seeing a lot of genotype by environment interaction, and what we were doing was we were misrepresenting the frequency of environment type 1 in the target population environments and undersampling it in the multi-environment trial. So what we were then interested in doing was thinking about using these managed environments to try and increase the frequency with which we sampled this multi-environment, this environment type 1 in our multi-environment trial to more consistently move up into these sampling situations where we were getting a high genetic correlation between our multi-environment trial testing system and our target population environments. And then in that situation, we were improving our confidence that the data we were analyzing in our multi-environment trials was relevant for the target population environments. And then you can start to think about how you can focus on some of the other components of the breeder's equation to accelerate rate of gain in your target population of environments. Just to give you a little bit of a look of some of the analyses that underpin this. Again, this is another way of looking at the data, the empirical data behind this response surface, where instead of looking at environment types, I was just looking at for the different managed environments that we could actually set up. If we were looking at 1 or different combinations of 2 of those, or different combinations of 3, right up to different combinations of 9 different combinations of different managed environments that we were setting up. And then looking at the genetic correlation between the yield data we would get in these combinations of the managed environments and the on-farm target population environments. And obviously you'd want this to be high. And what this actually shows you is that it is possible to come up with combinations of managed environments that are give you a high genetic correlation to your on-farm target environments. But it's also important to realize that you can actually create combinations of these managed environments that give you poor representation of the target population environments. So there's nothing magical by themselves, these managed environments. What you've got to do is get the right mixture and frequencies of these managed environments to allow you to see genetic variation that's relevant to your on-farm target population environment. So then you can start to think about, as I was discussing here, emphasizing the right environment types and trying to get their frequency of occurrence in your testing system high enough that you can be predictive of your target population of environments. So the concept is pretty straightforward, but what this, hopefully this distribution of results actually shows you is that, unless you're really committed to working out how to design and consistently create the right managed environments, you could often generate data that was unrepresentative of your on-farm target population of environments. So sort of moving along, we were interested, and this was work that I started with Greg Edmeades when I joined Pioneer. We were really interested in whether we could establish a set of managed environment testing systems that could support the maize breeding work we were doing for the western part of the Corn Belt. So in the same, using the same principles, but this time with more resources and more capacity to set up stations and support those, we were looking at establishing at different locations, design managed environments that would create consistently environment types that were representative of drought when it occurred in our target population environments. And so this is just a large multi-environment trial that was run by the business teams, where we were testing genotypes throughout the Corn Belt. The red dots just represent wherever we got an actual water deficit. But then within these water deficit environments, you can apply exactly the same principles that I talked about for wheat to characterize the timing and intensity of stress relative to flowering time. So we can start to sort out these environments in terms of whether they resulted in flowering stress or grain filling stress or combinations of those. And the green dots would be your water sufficient environment types where you didn't suffer water deficit. There were a lot of other experiments that were conducted where other factors were important, but these are the subset of the environments that we sampled where we were looking at contrast. So water availability on a continuum, and this picture down here at the bottom just shows the continuum of water availability on the x-axis, the yield variability on the y-axis, and the vertical distribution in here would be indicative of these different severities and timing of stress that would result in yield decrease relative to the potential if you didn't have any water deficit and you were mostly sampling the water sufficient environment types. So again, the same framework, now just applying that to maize for the US Corn Belt target population environments. So going into a little bit more detail of just some pictures here. Yes. In question? No. And just looking at some of the detail in some of these experiments where we were setting up these managed environments, we spent a lot of time at different stages of a maize breeding program. So here's just a cartoon of a maize program where we've got our heterotic groups, the females and the males. We're running basically pedigree improvement within a reciprocal recurrent selection framework. So we're developing improved inbreds on the female and on the male side in experiments. These blocks down here just represent these experiments, and also using those inbreds to develop hybrids and test those hybrids. So both of these stages of the breeding program could be designed and evaluated in different environmental situations at some of these stations where we set up the infrastructure to design and execute these managed environments, generating a range in water availability, which represents this distribution of evapotranspiration in the target population environments. Then we could manage the timing of that water availability to generate these different types of stresses that occur at these red dots, so that we could start to investigate how different hybrids would perform in these different types of conditions. So this is just a demonstration experiment that we actually ran, where side by side here we're using drip tape, where a drip tape line runs with each of the rows of the plots, so we can manage the water carefully within these plots. This would be your water sufficient, well-watered full irrigation experiment. Here the water deficit was timed to impose a grain filling stress, and here a flowering stress. And you can see some of the height reduction that happened here, less so in the grain filling stress relative to the fully irrigated. Then from within this demonstration experiment here, there were the series of hybrids, some that were tolerant to water deficit, some that were sensitive. And here's a contrast for two hybrids in the well-watered treatment here. So they both yielded quite well. There were similar yield levels, typical of very favorable conditions in irrigated situations in the western part of the Corn Belt. Here is a contrast under grain filling stress, where you can start to see a bit of a difference between the tolerant and the sensitive hybrid. And then in flowering stress, where we imposed the water deficiency right at the time of pollen shed and silk emergence, you end up with this more tolerant hybrid that managed to yield about 10 tons per hectare. The sensitive hybrid basically was barren and no yield basically from that experiment. So we could, with our management practices at these managed environments, we could consistently impose these types of stresses. You could do this in these demonstration experiments, but we could also start to do this in these larger scale breeding experiments. The other thing we investigated, and this was a lot of the background work, here we're looking at, on the x-axis, a whole range of water availability. So you can think about this as the quantity of irrigation that was actually applied. And then what we would do is we would change the timing with which we imposed the water deficit. And you can see in these environments where we reduce the amount of water availability, we end up with this large drop in yield relative to the potential. This dashed line up here would be for your very favorable, well-watered, irrigated experiment control. And what happens is if you get caught at flowering time, all of the hybrids suffer a yield hit. The more tolerant hybrids, less yield loss. The sensitive hybrids lose a lot more yield, and you get this contrast here. We went from somewhere around about 10 tons per hectare down to about 6 tons per hectare. So not quite as dramatic a yield loss as the picture I showed before, but still nevertheless a significant yield difference. And these were two hybrids that were being grown on more than 1 million acres of the Corn Belt. So it sort of shows something like of the yield contrast that could happen in the target population environments. So that we knew that we could actually design these managed environments to impose these types of stresses. The challenge was then to do it at the full scale of the station and to do it for multiple breeding programs. And this was all part of building up the team to support the design and execution of these managed environments consistently year after year across multiple locations. And then if you can do this consistently, if you think about these early stages of the breeding program here, over time, you can build up sort of very large meta data sets that can then be used to do a lot of trait mapping, and they can also be used to build training data sets for genomic prediction. So this was the starting point around which we actually designed combinations of the managed environments, which are these red, orange, and blue colored sort of rectangles here in our G/E matrix. Then the green are the on-farm testing that we did alongside this. So we never completely replaced the on-farm testing. We were just trying to improve the reliability with which we were sampling key environment types that occurred in the target population environments, when in some years you might undersample some and oversample them in other years. And then the important thing was also to make sure we had an appropriate set of checks that allowed us to determine when we were imposing the stress that allowed us to separate the tolerant from the sensitive maize hybrids. So building the team and the infrastructure to do that was quite a lot of work. And as I mentioned, this was work that was collaborative with a large team of people, beginning with Greg Edmeades and some of the people that he employed, and then the teams that we developed from that starting point. And so just to give you an idea of how we could actually use this large meta data set to build different training sets, we then use these data sets to train our genomic prediction models. And here's an example of just an analysis of one of those configurations of these data sets. And so this is a similar picture to what I showed you for wheat, where we're looking at different numbers of managed environments along the x-axis here. So these are different numbers of combinations of different types of managed environments. And to give you an idea of what these different numbers may represent, if we go back to this picture, they could be different timings of water stress and severity of water stress that we're sampling down here to impose different variants of water availability. So we're trying to sample not just a specific type of water stress, but we're trying to look at this whole distribution of situations that can actually occur on farm. And so that's why we're looking at these different numbers of locations, so that we can actually get some confidence that we're not overfitting to an underrepresentation of the target population environments. So for each one of these numbers of managed environments, there's two box plots here. The upper aqua-colored ones are for the checks. So the checks were included in all of these experiments here, and we had our sensitive and tolerant hybrids inside the check set in all of these experiments. So what we were using these for, as well as references for evaluating all of the new breeding material, they were also used to help us to calibrate when the managed environments were actually executed by the teams adequately. And so our target was to try and ensure that we could separate our checks consistently with a correlation of around about 0.8. So this was the target that the team was always aiming for. And as you can see, you get a whole distribution of results. And this is where you have to do a lot of quality control of your data sets once you've generated them. So even after all of this work, if our check correlations fell off of our targets, we would actually wait or remove some of these these environments from our data set. So this helped us to actually learn how to increase the representation of the target population environments with our managed environments. The lower box plots here are for the new entries that were coming into the breeding program, where we were trying to predict other drought environments. So this is the predictive accuracy that we were able to achieve in this particular example, where we were looking at predicting at this early stage in the breeding program. So you can think of that as your TC1 or top cross 1, predicting new genotypes into new drought environments, but still within the same general environment type that was a target for the target population environment. So this might be the severe flowering stress, for example, environment type, and that was the level of predictability we could get for the new germplasm that was coming into the breeding program, just as an example. Then sort of from these large data sets, another aspect that we spent a lot of time looking at was the correlation between these experiments and when we ran the same experiments in the target population environments. And we were able to do this, if you remember back to this cartoon. So this prediction that I'm looking at here, where we're looking at this predictive accuracy part of the Breeder's equation, relates to analyses within these managed environments down here. So you could think of this as being all inside the dark red or the orange set in here. And so now what I'm looking at is the correlations from these same data sets between the different environment types in the managed environments to the green or the on-farm execution of these experiments. And this is an example that's at sorting out the family level. So there's 100 or so different families that we're trying to sort out in this particular experiment. And what you can see is that these are the genetic correlations that we were able to achieve on the female side in this particular example between the water sufficient environment type, the stress environment type, to the comparable environments inside the target population of environments. And this was for the female side and the male side of the breeding programs in that case. So the correlations, these correlations between your managed environments and the target population of environments, and that's a breakout of the whole data set, were positive. And so then you could make your judgment whether or not this was acceptable relative to other investment of resources or how you would try to improve your realized gain on farm. The other part of this, which is sort of often not as heavily thought about, a lot of people think about these correlations here, but I was also very interested in what traits and what sort of genetic variation were we actually exposing in these different environment types when we were actually seeing genotype by environment interaction. So this is another way of thinking about this classical picture that people often have when they do reviews of G by E interaction. If you think about this as two environments down here, you're measuring a trait here, you've got two genotypes. Top picture says there's no G by E interaction. Here, this picture here, we've got heterogeneity of genetic variants. Here we've got crossover interaction, and here we've got a combination of crossover and heterogeneity of genetic variants. And so these interaction types, instead of just being a cartoon, may actually be the types of interactions we see when we're comparing the different environment types along this continuum of water availability from drought through to water sufficient. And then you can start to analyze the different trait combinations to the yield variation you're seeing in these experiments. And so Charlie Messina developed a capability around these experiments to actually look at how the different traits were contributing to the yield variation inside these experiments. And so the two things that I'll highlight in here, so you can think about this as a continuum from water stress to water sufficient. And then you've got these different colors. The size of the area under for the color indicates that for that particular trait on the legend here, that's the amount of variation for grain yield that trait would explain in that environment type. And so what you can see is that in the stress environments, you get some traits that are making strong contributions. When you go to the more favorable environments, they're making less contribution, and other traits are making a stronger contribution. The other feature that I'll show you in here that's really important is this darker blue area is all of the conditional trait by trait interactions that are happening. And so what you see is that when you move away from the extremes, so a lot of people spend a lot of time only looking at G by E interaction between these extremes, and then they study traits for drought stress only in these conditions and get worried about the G by E interaction to the other extreme. But most of the target population of environments is in this middle space here, where the trait by trait interactions are really important. So that was the other aspect that we spent a lot of time using our managed environments to investigate, were combinations of traits that we actually had to select for inside the breeding program. And that understanding then underpins how you weight different traits as they would contribute to your index for your selection, whether it's on phenotype or genomic prediction. And so just as one example, inside these managed environments, one of the traits that's on this picture here, this limited transpiration trait, which makes a contribution, particularly under the water limited environments here, but it also makes a very strong contribution to this interaction space here. We were interested in if we were to select for some restriction on transpiration, and actually in high vapor pressure deficit environments that can occur under the drought conditions in the western part of the Corn Belt. If we saved some water under high vapor pressure deficit conditions, would that be enough to contribute to yield improvements under water limited conditions? So we started off doing this very theoretically, and then we were again in these experiments developing a lot of in-field sensor capabilities to measure transpiration at the canopy level. And what we were able to demonstrate was that if you were to study some of these sensitive and non-sensitive hybrids, they were significantly different for this rate of transpiration at the canopy level, at the field level. And so in this case, this was one of the hybrids that was selected to restrict transpiration. And you can see that this restriction actually reduced the amount of transpiration. So in this day, there's about 1 millimeter difference of water use, which doesn't sound like much, but that's cumulatively for 1 day. And then if you accumulate that in these experiments over the course of a season, cumulatively over days, it can result in a difference. And so here again, the dark dot in this case is the hybrid that's restricting transpiration and conserving water, the solid line. There's more water available in the soil by the time you get to flowering time relative to the hybrid that's actually using more water through transpiration. And that difference is about 25 millimeters at flowering time, which can be a significant amount of water if you're under severe water deficit. And when we look at the water availability in severe water deficits at flowering time, this difference in water use can actually explain a large fraction of this variation that we see in the hybrids. And again, at the larger scale, this is the percentage, the variation for these traits that's contributing to this yield variation. So this one trait doesn't explain all of this, but it explains a fraction of that. And then you can start to look at over your whole target population environments. In these water limited environments, what sort of yield difference might this contribute over a wide range of the different types of water deficit environments? So it doesn't contribute everywhere, and there's a slight yield penalty at the top end, but in these water limited environments, you get a significant cumulative yield advantage. So what I sort of hopefully done is done a quick tour around some of the background work and tried to emphasize that it's a pretty long journey of how you actually develop and a technology like this and put it into practice in a breeding program. So I'm going to stop there and stop sharing, and hopefully I didn't go too long, and I'll hand it back to Dorcas.
Thank you very much, Mark, for that nice presentation. I would like to open the floor to questions at this moment. Please just raise your hand and then I'll give you the floor. Yeah, as we wait for people to ask live questions, I think we had a question in the chat.
You want me to look in the chat?
Sean, could you provide an estimate of the proportion of phenotypic variance attributed to G by E interactions, genotype by year, genotype by location, etc., that would justify the establishment of managed environments?
Yeah, so I would say a rule of thumb is you could look at it in terms of variance components, but I think that anybody that sort of makes their living out of doing this type of work is more interested in how they translate into the genetic correlation between your testing system and your on-farm target population environments. I think every breeder should be measuring that. I measured it all the time in my breeding program. Whenever that genetic correlation drops below 0.8, you're probably getting to a point where you better start asking questions about whether or not you're on target or off target of your target population environments. So anybody that understands the variance covariance framework around which we estimate variance components, the G by E interaction component is a mixture of the heterogeneity of variance and the lack of correlation scaled by those variances. And so that's why I emphasize that, you know, you could have a lot of G by E. Some of that could be heterogeneity of variance. So I would explicitly look at the genetic correlation component. And if it drops below 0.8, you should be starting to ask some serious questions. Whether or not that's by location or by year doesn't really bother me, because they're just really different ways within which you sample environment types, in my experience, in all the crops that I've worked with. And then you can also think about how agronomic management fits into that as well. So this whole thing fits into a G by E by M framework.
Thank you, Mark. I'll now go to Ephraim and then Maina Sarah. Ephraim, please.
You'll have to unmute, Ephraim.
Okay, can you hear me now?
I can hear you now, Ephraim. Yep, go ahead.
Thank you. Thank you. Hi, I think this is a very nice presentation. I have 3 short questions. The first one is about how do you compare the environmental types with the conventional environmental indices? The second question would be, to which extent, according to your point of view, the managed environment that you showed represented the TPEs? And the third question would be, from some slides you showed, I've seen that susceptibility to stress was mostly related to seed setting. And then my question would be, was it a failure of the male or the female organs? Thank you.
Yep. Okay. So you asked 3 questions. So I got to make sure that I remember to answer all 3. So the last one that you asked with regards to whether or not it was the male or the female. So in the drought, typically what I found, I would say, is that a lot of the variation for the traits was contributing through damage to the female organs, rather than through the pollen damage. And so we actually, Chris Inselmeyer, who was working at Pioneer at the time, introduced me to a really nice set of experiments where he actually did controlled pollinations of very viable pollen onto damaged silks under stress, and even fresh looking silks, when you put fresh pollen on them where you'd kept away the damaged pollen, still were not viable. So there was certainly a strong element of damage in the maize case to the female organs. And for the wheat, I did not do similar level of experiments. So I'm not going to comment on that, since I don't actually have data to support the comments. The other second question, working back to your first question, was to what extent did the managed environments represent the target population environments? So my answer is that's explicitly calculated as the genetic correlation between the traits as they're measured in the managed environments and in the target population environments. And hopefully what you can see is, by the extension of the breeder's equation and with mixed model capabilities, if you run experiments on farm, you can explicitly estimate the genetic correlations. And I would argue that any breeder that's not doing that's not doing their job. So I would always estimate that. And in my case, depending on the aspect of the breeding program I was working with. That could be anywhere, as I showed, but genetic correlations of about 0.3 up to 0.8 to 0.9. And that was for stress. And one of the main reasons that we could actually accelerate breeding wasn't because there was any magic in the managed environments. It's just that you could easily have 2 or 3 years in your breeding program on farm where you had rainfall that meant you never saw stress. So you got 2 cycles of testing where you could never apply selection pressure. But because we were running our managed environments in the northern and southern hemisphere, I was doing 2 cycles of drought testing every year. So that, I mean, even if you got lower correlations, because you can sort of accelerate the overall cycling of your breeding program, you're getting an advantage flowing into the rate at which you're turning over your germplasm with confidence with the selection. So you've got to think about the whole breeder's equation when you actually look at optimizing. And well, I guess this is the group that actually does a lot of this work. So you guys all know that, and you're using this sort of framework. Then you asked the other question about how this might relate to the classical stability indices. I mean, I've never been a great fan of the classical stability index or the sensitivity or indices of the environments in terms of mean yield, because I can get to the same mean yield from many different ways. And that's exactly why I showed that if you've got more explicit understanding of availability of environmental variables such as water and timing of that water availability, you get a discrimination of, even at the same yield level, you know that they're different environment types. So there's more information in there. So you'll get some coincidence of a classical environmental index, but the classical environmental index is a very coarse-grained environmental characterization. These tools that I demonstrated are much more fine-grained and give you more resolution of sorting out the environmental contributors to G by E interaction. And then you can decide how you weight that information with that, with empirical evidence from your experiments. I think I managed to cover all 3 of your experiments, Ephraim.
Yeah, great. Thank you, Mark. Great to hear you again. Thank you.
Sorry, Maina, please go ahead. Maina Zahara, can you hear me?
Yes. Sorry. I had a issue with my mic. Mark, many thanks for the wonderful presentation. Mine is a quick one, is related to the transpiration restriction that translates to water conservation.
Yep.
I did some work on that in the past, and it's relatively easy to phenotype for that under growth chamber conditions where you can play around with the VPD. Have you tried anything of that sort in a field setup?
Yep. So no, you're absolutely right. So a lot of our early work in playing around that was in chambers where we could manipulate VPD. So exactly as you said, and we actually collaborated a lot with Tom Sinclair on that work, and he gave us a lot of tools and capabilities to actually do that initial phenotyping. In the field, one of the ways we tried to scale this up was by going to different locations where we knew we would be able to get high VPD in the field, in the western part of the Corn Belt, and in some of the drier areas, such as in California, where one of our managed environment locations was at Woodland, and also at a high VPD site in Chile, where we could get that high VPD condition as well. So we really moved around across different locations to get a gradient of different VPD situations with different soil water supply situations. So wasn't as much control as you would get in a control chamber, but that was also why I was trying to emphasize we tried to sample a range of replications of these conditions to scale this up to breeding program level. So that was the approach. And yeah, it's sort of taking a lot of the detailed knowledge from the physiology work and trying to scale it up and make it and provide useful information for a selection decision inside a breeding program. So hopefully that at least gives you an idea of the approach that I took, but we could speak for hours on this topic. I'll stop there.
Thank you. Thank you very much.
Before I go to Tokuma and Keser, there is a question from Yosef in the chat. Have all the testing locations been characterized for moisture deficit, or are there representative locations among the testing sites?
So all of the locations where we were running my breeding program, we did additional work even on farm conditions to characterize the levels of water deficit. So yeah, we would have automated sensors, and sometimes initially individuals would spend a lot of time going to those locations and actually taking a lot of those additional measurements so that we could ground truth that we were in fact getting the environmental conditions that we were seeking, and then we had confidence in the data. The other thing that I would emphasize is it's not unlike many other situations. There's a lot of times that you execute an experiment like this and it completely fails. I can remember one experiment I ran where I think I had something like 6,000 yield plots in the field, and the stress was so severe. We did a really good job of measuring transpiration, but it was so severe, every single hybrid had 0 yield. So I mean, it takes a lot of persistence and patience. And I guess what I'm trying to emphasize is that just because you're working on managed environments, there's many ways you can generate bad data with managed environments, just like you can get a bad sample of locations and years to represent your TPE as well. So you got to put a lot of work in to make these things relevant for your target.
Yeah, thanks, Mark. Tokuma?
Thank you, Mark, really, for your excellent presentation and sharing your experience. So maybe if my question is related to what you have, I want to know from your extensive experience for the environmental characterization from your presentation, I have seen it. So what really the character that we have to be looking for during environmental characterization that can help us for the G by E by M experiment? I mean, is that like a water deficit? In which character we can come across on it? Because most of the time we are looking for like annual rainfall, altitude, and something and so forth. So what are really the basic character what we need to use for environmental characterization before doing the mega environment experiment? If you get me my question. Thank you.
Yep. So, I mean, I think that sort of is a really big question, and it's probably would be interesting to get sort of a lot of perspectives from this group. My experience was that the weather data today is relatively easy to get better quality weather data, certainly than when I started. The thing that I still find that's the hardest to get good quality data for are soil characteristics that influence the total water holding capacity of the soils, I mean soil depth, information, and in many cases, the soil maps that we started with were very poor indicators of the soils at our experimental location. So we had to either go and characterize the soils ourselves and correct some of the information that we had in the soil maps. But even then, we had to spend more time just understanding specific features of some of the soils to influence water availability. Getting reliable data on temperature inside your experiments, I think, is something that's really important, particularly when you're actually trying to interpret the interplay of sort of hotter and drier environments. And so making sure you get good temperature data with that water availability is important. The other thing, there's some basics that it always surprises me that people get wrong, like planting dates are sometimes wrong, plant populations are wrong. It's amazing to me how many times people don't record when they irrigated their experiment. So in many cases, when we were trying to ground truth data, we actually found out that someone irrigated when they weren't supposed to irrigate and so forth. So there's a fair bit of work that's involved in that. And so you've got to dedicate some time. You can't assume that people are always doing what you think they're doing, I would say.
Thanks, Mark. Keser?
Thank you, Mark.
Keser?
Yeah, thank you. Yeah, thank you, Mark. That was a great presentation and interesting. At the end of the day, after you make the selections, you make the germ, you select the germplasm that fit or adapt to environments. So by doing those manage the environment tries, that making the selections, whether your germplasm is being led towards the wide adaptation or specific adaptation. If it is wide adaptation type of the germplasm you have been developing, how wide it is, or if it is specific, how specific it is. Thank you.
Yep, good questions. So I would say that it was a bit of both. And so the first few cycles of breeding that we were focusing on were really trying to improve broad adaptation by removing some of the major weaknesses that some of the maize hybrids had, because they'd really only been selected under very favorable environments. So the example that I showed you of the hybrid that actually had zero yield under the flowering stress, that hybrid was being grown on millions of acres in the US Corn Belt. But it had been developed for irrigated conditions, and then people were starting to grow it in limited irrigation and dryland farming, where it was obviously increasing their risk if they didn't get rainfall. So that was an example of where we tried to work with that germplasm to give it traits like limited transpiration that broadened its adaptation. But we could actually go to the extreme. If you were working with dryland maize farmers, you could actually select very aggressively for specific traits and improve specific adaptation. For the commercial targets that I had, those types of hybrids weren't the major types of hybrids that a large commercial company was trying to develop, where there were interests in broad adaptation. But for the smaller companies, those types of hybrids could be a significant part of their portfolio. So with the knowledge, it's possible to build both. But again, you've still got to live within the biological constraints. Some traits are going to help you a bit with broad adaptation. So the limited transpiration helped with that. But if we went after very aggressive silking in drought stress, then that could put a much more severe limitation on yield potential, and it would be more specific adaptation. So you could actually pivot and develop both of those types of hybrids. So I think your point's really good. I mean, I think that none of this solves your responsibility for ensuring that you're developing hybrids that meet farmers' needs. And it's not about developing one hybrid that works everywhere all the time, in my opinion.
Yeah, thanks, Mac. I'm sorry we over time, but I do have 2 questions in the chat. So could I ask you this, or would you prefer that I send you via email?
You're fine. I'm already way past my bedtime. It's after midnight here, Dorcas, so I'm going to sleep well regardless. What were the other 2 questions? Go ahead.
So the first question is, in crops without synchronized flowering like bananas, what would be your recommendation on using managed environments to capture the effect of environment on genetic gain?
It's an easy question for me to answer. I don't know. I think that you are going to have to undertake your own research like we did for the grain crop. So it's an interesting question. I don't know the answer, but I think the same framework of asking this question is something you should think about. And hopefully what I've done is, I've got a Scottish background, so I'm not a super optimist. I mean, this is hard work. I mean, it's not like any of this is sort of a free lunch. I mean, you've got to work hard, and I can only imagine that it's going to be a bit of a challenge if you do it for some of these other crops.
Yeah. Well, thank you, Mark. The next question is, does the correlation between TPE and managed stress environments would be same in both field and controlled screening?
So I think so if you mean by controlled screening on field, do you think the question means in like a controlled environment chamber or something, Dorcas? I'm just trying to make sure I'm answering the right question.
Yeah, I am assuming it's controlled like in a screen house, but Matthew, are you online? Would you like to ask the question live? Matthew, Anurag? But in this case, I assume that he's asking about the field as well as maybe screen house or greenhouse screening.
Yeah. So for some traits, I think you can achieve equivalent correlation between the screen house and the field for your target environments. But for other traits, that's going to be extremely difficult to do. I think that you really have to look at how representative all of the setup in your screen house is for your field situation. But again, these are very empirical questions where I think you have to go in and actually estimate the appropriate statistics by running the right experiments.
Yeah, thank you, Mark. I want to thank you for taking this time. Claudia, you have a question on where you get the recording. I think my colleague Julie is still online, and we will be sharing the recording of this presentation and the previous ones as well in a page. So thank you very much, Mark. Sorry for going over the time, but we do appreciate your willingness to talk to the team and generate this whole enthusiasm. So
happy to do that, Dorcas. Nice to meet everybody again, and I'll probably meet some of you again in the not too distant future, all going well.
Thank you, everyone, and thank you for staying over time, and see you in our next occurrence of the COP.
Okay.
Thank you.
Bye.
Bye-bye. Thank you.
Bye-bye.
Fine. Bye-bye.
Yeah.